They are often dismissed as monsters of the sea, but these majestic, enigmatic creatures are actually essential wardens of our marine environment. Found in every ocean across the world, they play a fundamental role in ensuring balance and stability in our oceans. Sadly, this role is jeopardized by overfishing and habitat loss. Around 3 billion people rely on fish as their primary source of protein. Ongoing declines in shark populations could have devastating and unpredictable impacts on marine food webs and even our own food supply. The plight of sharks and rays was the spark for the Save Our Seas Foundation, founded over 15 years ago. Through the funding and support of research, conservation, and education initiatives, we are dedicated to safeguarding the future of sharks and rays. Today we have 3 centers based in Seychelles, South Africa, and the USA. Since 2003, we have funded over 300 projects on over 60 species in over 50 countries. We have also partnered long-term with several projects, including the Shark Spotters in South Africa, the Shark Lab in the Bahamas, and the Manta Trust. We're passionate about our mission: protect the future of sharks and rays and our oceans for generations to come. We can and will make a difference. The Save Our Seas Foundation.
I used to actually be terrified in any water where I couldn't see the bottom. Tales of monsters lurking in the deep. What are these enigmatic, crazy creatures? The first recognizable ancestors of sharks existed before trees, whereas now they could be facing collapse in a single generation. I actually think it's very hard to know just how much you love something until you lose it. Grief is the price we pay for love. The thing that drove me on this path was a pure passion for the animals themselves. I didn't start out wanting to save them because I didn't know that they needed saving or help. This one's for the ocean. I realized that if we want to help save sharks, we need to make sure they have safe places to live in.
Hello, everyone. Welcome to the Save Our Seas Foundation Distinguished Speaker Series. My name is Lance. I am the Director of Sustainability and Workforce Readiness at the Museum of Discovery and Science in sunny South Florida. We're so happy to have everyone here tonight. Thank you for joining us. This is our first episode of the 2025 season. We have a whole series planned out for you going all the way through December. So some of them we plan on at least one a month, sometimes twice a month. To see the whole series, you can go to our website at mods.org, mods.org, and learn about the whole series and sign up and hear all about these fantastic ocean researchers and conservationists and educators throughout the whole year. So keep in touch with us. Thanks again to the Save Our Seas Foundation for making this whole series possible. We really appreciate their support. and not only supporting us at the museum, but also supporting all of the research and all these conservation efforts all around the world. We hope to bring you the breadth of that support from all over the world through the series. From tonight, talking to Dr. Taryn Murray from South Africa, all the way to Asia, to our backyard in South Florida as well. So we're bringing you lots of different perspectives over the series this year. Just a few little things about our live broadcast tonight. We welcome questions. If any of you have questions for our researcher this evening, please feel free to ask them. We're going to ask that you put them into the chat. I will be able to see them, and I will ask them on your behalf. So please feel free. Don't be shy. Put your questions in the chat. We will have some dedicated time at the end for some like Q&A, but I will be able to chime in on your behalf here and there and kind of mix in what questions you might have. So please put them in there. Ask lots of questions. That'd be great. With that, I would like to introduce our speaker for this evening. Dr. Taryn Murray is an instrument scientist at the South African Institute for Aquatic Biodiversity and a research associate at Rhodes University there in South Africa. Dr. Murray's journey into marine science began kind of unexpectedly. She had really not a solid plan to study fish until a friend pushed her to take a course in ichthyology, the study of fish, in her undergrad. This revealed a passion she didn't even know she had. That decision launched a remarkable career that now spans over 16 years, during which she has explored illegal fishing in protected areas, studied genetic structures of reef species, and tracked fish movements through cutting-edge technologies. Today, Dr. Murray leads the Acoustic Tracking Array platform. This is a vast network of acoustic receivers deployed all across the 2,200 kilometers coastline of South Africa. This platform connects data on the long distance and long-term movements of marine animals, including fish, sharks, rays, turtles, and supports both national and international collaborations. Her work focuses on key species of ecology and fishery importance, contributing directly to the development of management and conservation strategies. Recent projects include mullet species tracking in the Eastern Cape estuaries, as well as movement of sharks and rays in False Bay, in partnership with organizations like the Shark Spotters and the Two Oceans Aquarium. Beyond her technical expertise, Dr. Murray is also a passionate mentor and is committed to training the next generation of marine scientists. So, Dr. Murray, welcome. We're so happy to have you.
Thanks very much for having me, Lance. Very excited to be here.
Yeah, we're excited to have you. You've already shared your screen, so that's fantastic. Just a reminder for our audience to put in questions as we go through. Take it away. The stage is yours. Tell us all about what you do.
Thank you very much, Lance. Hi, everyone. Thank you so much for taking the time to join us, for me anyway, the very early hours of this morning on Friday morning. But it's great to be here.
It's midnight your time, right?
It's midnight, yes.
Thanks for staying up late for us.
No problem. I always love sharing what we do, and I get very excited, and I'm very passionate about sharing the incredible work that not just myself, I really can't take credit for it. It's really a South African team effort. So I'm hoping to share some of that with you this evening, and I hope you enjoy it. So people have been interested in tracking the movements of multiple different animals, right? You've got birds, you've got turtles, you've got wolves, you've got all sorts of animals. And these include tiny things like insects. I mean, people can even put little tags on insects now, and you can figure out where, following these incredible migrations of butterflies all over the world, So I did a bit of a deep dive for a talk a while ago, and I found out the first insect that was actually tagged was in 1920. That's over 100 years ago that people have been studying the movements of insects. When you look at the birds, they really are the top dogs, or should I say the top aviation species. They definitely get the gold medal. They've been tracked, or at least have had some of their movements studied since almost the 1600s, which is just absolutely incredible. But studying animals on land is relatively straightforward because a lot of the time you can keep your eyes on them. But what about animals that you can't see all the time? What about those things that live under the water? So what I'm hoping that we're going to do this evening is really hold our breath and take a deep dive under the environment where we can't follow animals all the time. So when we think about fish tagging or monitoring the movements, figuring out where fish and aquatic animals move, how do you do it? How do you go about this? I mean, it's not really any different to tagging anything else. You would place some kind of tag on it, but when did this all first actually begin? So some guys in Scotland, some wealthy landowners, they had a river on their land and they thought, you know what, we've got some salmon, we've got some trout. What do they do? Where do they go? Because I guess through fishing, they figured out that sometimes they were there and sometimes they weren't. So they tried to tag them in some way. And this might have been as simply as cutting a little piece of their fin or something. But then they could figure out, oh, okay, these animals are actually moving around. So this happened over 150 years ago, which still just blows my mind. So it definitely pays to be curious and to figure out ways that you can answer these questions that you have. But since then, the types of tags and the way to study movement behavior has absolutely exploded. So first of all, what is tagging, right? There's so many different tag types, but what is tagging? It's basically making a fish identifiable in some way that if somebody else recaptures that fish, you can recognize that individual. So you can get information on all sorts of aspects of that fish based on how it's tagged. The fish itself has actual natural tags if you want to monitor it throughout its life history. This, for example, are the ear blowns or the otoliths of a fish. And they essentially lay down rings of like calcium deposits. And through figuring out or through counting these rings and for actually you can take sort of the chemical composition of these otoliths and you can figure out where animals have moved throughout their lives, which is really interesting. But unfortunately for that, the fish might have to come to an end, but it still provides a really amazing amount of information. You can then start looking at things like genetics. You know, you can figure out if I catch a fish over here in South Africa and it maybe moves all the way to the Comores or Kenya, and you take genetics of an animal over there, are they the same? Are they different? Is my fish very similar to the ones in Kenya? So that also starts being able to answer some kind of questions into how fish are moving. You can take something as simple as a fin clip. So you make a little notch in a fin, and you then know that that animal has been tagged or identified in some way. You can get things called VIFE tags. So pretty much you take a little injection and you essentially inject like a fluorescent material under either the fins or some certain place in the fish's body. And you can then go back. This is mostly for like scuba or snorkeling surveys where you tag a whole bunch of fish and then you go and snorkel again and you're like, oh yeah, there's that fish that I tagged with 3 red stripes. So this is like more for studies that are really small, or if you release a whole bunch of fish and you catch them again and you can figure out you know, how many stay in a specific area. You can get dart tags. So these are, there are tons of programs that use dart tags around the world. South Africa has our own dart tagging program called the Oceanographic Research Institute Cooperative Fish Tagging Project. So it's quite a mouthful, but it's been collecting information on the movements of numerous different species in South Africa, sharks, rays, tons and tons of fish species using these plastic dart tags. And it's been in place since 1984. So we're in the 41st year of this program, which is just absolutely incredible. You get pit tags. So if you go to the vet, you take your animal, your cat or your dog to the vet, and they get a little chip in their ear. It's literally the same thing. So you can use those and figure out what animals are doing specifically in like small river systems. You get satellite tags. These are always the exciting things, but these mostly go on like your big sharpy, bitey, toothy things like your big sharks or your huge predatory fish like your marlin or your sailfish. And then you can also get your acoustic tags. And this is really where I'm going to be focusing my talk on, is acoustic telemetry and these acoustic tags. So first of all, what is acoustic telemetry? I'm just going to give you a solid little quick crash course. So it essentially consists of two components. The first is an acoustic receiver. So this is the piece of equipment that you deploy or you place in the river or the estuary or the sea, wherever you want to study the movements of your specific species. So in our case, we place our estuary or our receivers in estuaries in these nice cement blocks. And at sea, they are either moored down and then they're retrieved by guys that go down on scuba gear, or you get really fancy ones now that can basically release remotely, and then the whole thing just floats back to the surface, which is really great for areas that are maybe too deep to scuba dive, or areas that are maybe a little bit dangerous. So perhaps strong currents or something similar. So what does this look like? So in South Africa, our estuaries are exceptionally small. An estuary is where the sea and the river essentially meet. So you've got salt water and fresh water mixing in this area known as an estuary. So ours, as you can see, are wonderfully brown and muddy. It's always great swimming around, where when we have to go find these things, it's basically like wearing a blindfold and you can't actually see what you're doing. So you're just fumbling around in the dark, hoping to find your equipment again. So it's always a bit of a challenge, but yeah, it's good fun. And then at sea, you put them over the edge and there goes the unit down to the bottom. We leave them for about 6 to 8 months before we take them out the water again. And the ones in the estuary stay for up to a year. Okay, so we've got our receivers. They're in our estuary or river or sea, wherever we want to study our movement behavior. Then you've got a whole bunch of different fish and sharks swimming around in your area too, or in your system. And this is the fun part. This is what really attracts a lot of students, should I say, to our field, is the prospect of going fishing. So you get your fishing rod, you get your tackle, you go out and you actively target the animals that you're hoping to catch. So it is good fun, and people are always trained to handle the animals in a very good way, and the handling skills have really been honed down quite nicely. So the way that the researchers or students handle the animals is always really, really great. So you've got your little acoustic tags in your fish, but how do you actually get these tags in fish? Well, you have to do pretty much an operation. So you catch your fish. We generally put it in a tub with an anesthetic in it to essentially make it go to sleep. So it essentially just relaxes the fish, and fish breathe with a percula on the side of their by their gills. And when that sort of slows down and the fish kind of rolls over, you know that it's anesthetized enough to start the surgery. You do a little incision, you insert the tag, you stitch him up, and then he's happy to go on his merry way. These tags themselves actually come in all sorts of different sizes. So I've just used a standard highlighter, that you can mark in papers or anything. And the biggest one on the left can go for up to 10 years. So you can track a single individual animal for 10 years which is just unbelievable when you consider that some species might only live for something like 12 years. So you're essentially monitoring the whole life of that individual. You get really, really small tags now that only go for a couple of months, but those ones are in to tag kind of small juvenile fish. But it's just unbelievable the technological advancements that have happened over the past couple of years.
I'm sure battery technology has allowed these things to last a long time too, right? Lithium
Absolutely,
right?
absolutely. Yeah. And I think
incredible.
you know, the nice thing with the smaller tags is that they're starting to make batteries that are now smaller, allowing for smaller fish to be tagged, which has always sort of been a missing a gap in the knowledge. So that's like really exciting stuff.
Do any of the tags use like to power themselves? I often think of like watches. You know, you can have a watch that is powered by your movement, essentially. I wonder, are there any tags that incorporate that technology?
I'm not too sure of. There are tags that kind of incorporate a movement component called accelerometers, and that can actually start telling you how a fish is moving. So it's not just a case of, oh, it's here somewhere. It's a case of, oh, its head is down, or now it's swimming up, or, you know, actual changes in the movement, the angle, I guess, of the fish. But being powered by the movement of the fish, I'm not too sure, but that would solve all the problems. That would really solve all the problems.
Call Seiko, the watch company, making an animal tag.
Definitely. Are you interested in acoustics? We've got a business proposition for you.
Right.
So once you've tagged your animal, you allow it some time to recover. So if it's in an anesthetic bath, you need it to be able to recover fully before you release it back in an environment, because you know, if a fish is a bit drowsy and everything, it can probably end up not where it should end up, and you want to give it the best chance of survival. So you will go and you will this is a small little endemic species, so basically a shark species only found in southern Africa, called the spotted gully shark, and it's being released by one of our students, but he just waited there for a bit of a lull in the waves and then let it go on its merry way. So how the tags and the receivers actually work is, if you think of the receivers as a cell phone or a mobile phone tower, and the tag is your mobile phone. And the mobile phone can actually only work when it's in the reception, when it's in the detection range of that mobile phone tower, right? So it's exactly the same when it comes to acoustic tags and receivers. So once the little fish swimming around releases, the tag releases that signal, and the signal's picked up by an acoustic receiver, it records, the receiver records the unique ID code of the tag, along with the date and the time that that signal was recorded. So what you have to do as researchers, you take all these little pieces together, these little puzzle pieces, and you have to put together a giant movement puzzle. So that's basically what we're trying to do, is we're going to take all these thousands of different pieces and put this amazing puzzle together. So where did all of this begin in South Africa? It actually started with very humble beginnings, and it actually started with a fish. This is not the fish, full disclaimer, this is not the fish, but a fish named, affectionately named Walter. Walter was a dusky cob. So the photo that I showed previously, that is a dusky cob, and it is one of South Africa's most iconic angling species. They reach over a length of 2 meters or 6.5 feet. They weigh up to 80 kilograms or 180 pounds, and they can live for up to 42 years. But because they grow so big and so heavy, they really are targeted by the recreational guys. So these are the guys fishing for fun or for sport, and then also for the subsistence guys. These are the guys that are living for food. You know, they're fishing for food. They need to fill their tummies and provide for their families. So over the years, the numbers of dusky cob have actually dwindled quite badly because they are so heavily targeted and heavily exploited. if you liken to they spawn a biomass per recruit, it's a fancy science term for basically estimating how many individuals are left that are able to breed. And of all the fish, all the dusky cob that were once in South Africa, there are currently only 1.5 to 4.5% left. So I know it's like a ballpark figure, but if you look in the figure on the right with all the cob, that's pretty much 4 left. So if you have 100 fish, there are only 4 of them left that are able to spawn. So when you start thinking it and breaking it down in terms of those numbers, it gets quite scary. Because of this, they're listed as endangered on the IUCN Red List, and South Africa has actually developed a conservation strategy to stop the decline of the species. And it's not just this species, it's also 2 sister species called the silver cob and the square-tail cob as well. But back to Walter. Let's go back to Walter. So Walter was living in the West Kleinamonder estuary. So these are 2 sister estuaries that are literally right next door to each other. And South Africa has tons of what we call temporarily open closed estuaries. So a lot of the time, they're actually not even connected to the sea. And then suddenly the mouth will break open, you'll connect to the sea, and then it lasts for a couple of weeks or months, maybe even years, and then it closes off again. So this is what these systems do. But when they close off, fish get trapped in these systems and they've got nowhere to go. But the good thing is that these systems are filled with amazing things to eat for a big fish eater like the dusky cob. They love eating things like mullet, and these systems have lots and lots of mullet. So when a fish that is the size of Walter gets trapped in a system, you know he's going to be happy, you know he's going to get big. So what happened was that Walter was basically the guinea pig, and he started the whole years and years and years of estuary work on species living in estuaries. So Walter was tagged and he was manually tracked, which means people actually sat on a boat and followed him around literally non-stop for a week, figuring out what he was doing. So this was like a really cool, we're just dipping our toe into this world of acoustic telemetry in estuaries. But what it really showed was that we could study one fish from one species in one estuary. Since the pilot went so well, the next step was, okay, we're going to do a proper study now. We are going to tag 2 species in this system, and it was literally right next door to the Kleinamonder, to the West Kleinamonder estuary. This is called the Great Fish Estuary. And why it was chosen is because it had tons of dusky cob in it, and it's got tons of another species called spotted grunter that live in the system. And at the same time, the system is also heavily exploited by subsistence anglers. So these, again, are the guys who don't normally have formal employment or any employment at all, and they are literally looking for a meal. So they are providing for their families. I won't share the results from this work, but it basically shows that, yes, we can conduct a formal study, and it went amazingly. But what it showed was we can actually tag a lot of species of more than one species. I mean, a lot of individuals from at least 2 species, but still only in one system. As things passed, as things developed and time passed and years carried on, we became a little bit old hat at telemetry, but we branched out into other estuaries. We put receivers at the sea, so you could start figuring out all kinds of movements of these animals within the estuaries, between the estuary and the sea, along the coastline. Like, it really started growing so nicely. But through this, we were tagging lots of fish from lots of species in lots of different estuaries. But just to highlight the importance of this stuff, all of this work over the past, for more than 20 years, has actually been driven by 2 very dedicated researchers, and they are in the top left of your screen. They are Professor Paul Cowley on the left and Dr. Tu Nasher on the right. So Paul was actually my supervisor, my mentor throughout my postgraduate degrees. And then I was very fortunate to work for him. And very recently, he did stop working. And then Tur was, he is a researcher at the Norwegian Institute for Nature Research. And back in 2002, they had a crazy idea to put an application, a funding application, to get money in collaboration with South Africa and Norway. And let's just say that 5 years, we're now on our fifth call, 5 years, 5 calls, funding calls of this joint collaboration has really allowed us to carry on this amazing estuary work. And I'm just so proud to be able to continue this work for Paul and for all these numerous students that have come through the system, working on estuaries and using acoustic telemetry. So you might be wondering, why estuaries? Why are estuaries actually so important? And why were we like, you know what, we're going to do estuary species? Why not sharks? Why not rays? Why not turtles? Why not anything else? Well, estuaries are really, really important in the life history of so many fish species. So this pretty much means that you've got adults that spawn at sea. So they go out to sea, they breed, they lay their eggs, they do whatever they need to do. These little eggs then hatch, and the tiny little juveniles come back into estuaries, because this is where they stay for 1 to 3 years, because these estuaries are jam-packed with food. They've got safe places for them to live, to avoid predators. They are good thermal refuge places. So basically, if the sea's too cold, they can go into the estuary. If the river's too cold, they can go into the estuary. So they're really, really good nursery areas for these species. And then once their time is up as juveniles, they head straight back on out to sea. Some species, though, live for extended periods of time within estuaries. So this is why we really did this, why we tried to focus on these species. But it wasn't just the fact that these species themselves are important and estuaries are important. It's because estuaries are amongst the most modified environments that you can have, aquatic environments. You've got infrastructure development, which then leads to habitat degradation and deterioration. So for example, I saw in some of the videos at the beginning, you've got these really changed landscapes or changed waterways. Here, what I've pictured over there is the Kawi estuary here in South Africa. It's literally about half an hour down the road from me. And they pretty much developed this huge marina complex on a marshland. So they've changed the whole lower reaches of the estuary to accommodate people and growing population.
That's what South Florida looks like.
Yes. No,
yes.
That's why I saw those pictures at the beginning.
Exactly
I was like,
the same.
that looks very similar to our areas of, well, just some of them anyway. This is typically wealthier kind of estates that pop up. So these are very much not your average Joe day-to-day living. This is the big bucks. So these guys are wealthy people. But what happens in these estuaries is that because it's such a bottleneck in their life history, you've got small fish, you've got big fish coming to the system. They are so heavily targeted, with people often taking out more than what they should and taking out smaller fish than what they should. And this, I think, has largely been driven by a beach vehicle ban, which was implemented in 2001. So people were allowed to ride on the beach and they could fish wherever. But in 2001, the government said, no more, you cannot do this. And what it essentially did was take all that effort where all the fishermen were spread along the beach, and it dumped it on the estuaries, because completely stereotyping here, sorry if this offends anybody, I don't mean for it to be offensive, but fishermen are generally lazy and they don't want to walk 10 kilometers or 20 miles to a fishing spot. Why walk that distance when you can walk 200 meters down the road and you can throw a fishing line into an estuary? This actually also resulted in the complete local extinction of our sawfish species, which we had in higher up our east coast, so closer to the Mozambique border. We had a local sawfish species which has completely been fished to extinction. And many of these species are actually, the stocks are considered to be completely collapsed, so they are not sustainable in any way or very heavily overexploited. So movement studies themselves can be conducted on various scales. So I've placed a focus on estuaries. So by putting receivers in estuaries or in small coastal arrays, you can really start understanding movements within an estuary, between estuaries, little coastal movements. But when you start placing these huge curtains of receivers along a coastline, you can start understanding things like huge longshore movements or annual migrations. I mean, I'm talking, I don't know, 1,000 miles worth of migration. So it's quite extensive, the amount of information that you can figure out. So through all of this, all the estuary work, at the same time, this man over here, I mentioned him already, Professor Paul Cowley, he was in contact with our Canadian partners at the Ocean Tracking Network. So they've got pretty much a global network of equipment. And through talks with them and talks with our Norwegian collaborators, we have slowly over the years developed our very own coastal receiver array, which we've called the Acoustic Tracking Array Platform. And this covers, as Lance mentioned earlier, about 2,200 kilometers. I'm not too sure what that is in miles, from just north of Cape Town, which is down at the bottom left, all the way up to southern Mozambique. But I really want to take a moment to emphasize that this is such a brilliant collaborative network. It's not just something that it's not like the institute that I work for, we don't own all the equipment. This is really a joint effort of so many different researchers bringing their own equipment. And the role that I play and my colleague plays is that we essentially manage and maintain this network. So everybody joins the party and we not control the data, but we manage the data. We provide a service for the greater acoustic telemetry community, but very collaborative, and it's amazing. Everybody's very friendly. It's great. You don't often get that in a lot of places in the world, but everybody's on board and it's amazing.
So just for some context, I just want to say that, so it's about 1,300 and a half, 1,300 miles. That is equal to the entire coastline of Florida.
Yeah.
The entire thing, the whole peninsula. It's huge. It's a big area.
No, it is. It's grown slowly. When you still look at it and compared to the whole US East Coast, for example, then we're a bit of drop in the ocean, but we're quite proud of where we've managed to come, which is great. Oh, there we are at the bottom. So through all of this equipment and whatever else, you've got all these other incredible large-scale acoustic telemetry arrays throughout the world. I've stuck a whole bunch of logos up there with various networks across the world. But we're down there, the southern tip of Africa, holding our own, which is great. So what's actually tagged in the water? So over the years, we have obviously been tagging animals throughout the year, but to date, we've actually tagged almost 2,000 animals. And again, I say it's the greater community. It's not me and my institute, but everyone in South Africa has tagged just over 2,000 animals, which is, I didn't actually realize. I thought we were still sitting on about 1,800. So that was great to find out. And currently, we're monitoring 17 shark species, 13 fish, 8 stingrays, 3 turtles, 1 skate, and no penguins. We actually tagged there was an institute called Bayworld that tagged a couple of penguins, African penguins, but we unfortunately didn't pick up any of those tagged animals. So just jumping into some results, I literally do not have time to go through all the things. Otherwise, we could be here till about 5 o'clock tomorrow morning. So I won't put you through that, but just highlighting some really cool little examples. So as Lance mentioned earlier, we've been doing some amazing mullet work. I mean, who knew that these tiny little baitfish species would be doing such amazing things? So we actually ended up tagging 4 species in an estuary called the Kawi Estuary, which is the one that's half an hour down the road from me. And the reason we found out it was 4 species was because we actually ran genetic analyses on these animals because they look so familiar. We couldn't identify them confidently externally. So we're like, no, we got to get in the lab. We've got to make sure that these species that we're tagging are the species we say they are. So our 2 species became 4 species, which was probably expected, but a little surprising. But it just a quick little rough home range plot. It's really not accurate, but just to show that even though all these species live in one system, they still kind of do different things, which is really interesting. And we've got 2 PhD students that are working on this now, figuring out drivers behind the movements. Why are they doing different things? Do they work together? Like, yeah, all sorts of cool things. Then this is one of my favorite species, the Leophis. I'm a little bit impartial because this was the species that I studied for my PhD. In fact, that was one that I tagged for during my PhD, which is, yeah, very cool. So this is a little juvenile. This is a species called Leophis, and this is or leatherfish, if you want to directly translate it, Leophis. And this is a little juvenile, and we tagged them, and we also monitored their movements in one estuary. And we found out that, wow, you know, they're moving all the time, but they're actually doing some different things depending on each individual. So we had some individuals that were cruising up and down, and some that just spent most of their time at sea. We also found out that if we tagged them 2 estuaries, and we had receivers deployed in so many different estuaries, they were actually moving around far more than we ever anticipated for these small fish. I mean, these are fish that are about a ruler length, you know, and they're moving all over the place. So it was just absolutely amazing to figure out, or to find out, that these smaller fish were just basically estuary hopping. If you look at the adults, this is now a larger individual, a big adult. These guys, pretty much once they move out of estuaries, they start moving up and down the coastline. But when they reach sub-adult and adult life stages, they start undertaking an annual spawning migration up the East Coast. So they're moving thousands of kilometers, hundreds to thousands of kilometers to get to where they breed, and then they turn around and they come back. And this one individual was recorded down near where we live, and it moved up to, if anybody's, it's like the East Coast, mid-East Coast of South Africa, moved up to that place every single year. So now 2020 and 2021, there would have been movements of this animal up there, but unfortunately the equipment that was in the water at the time, the batteries died, so we weren't able to record that movement. But I can 100% guarantee you that that fish went up to that place 6 times and eventually met its demise. Unfortunately, that is the exact fish. But what an incredible story, because this animal was already an adult when it was tagged. So we pretty much recorded its entire adult life history, which is unbelievable. So this is the benefit of tagging animals with these long tags. Coming back to Walter, our friend, but this is not Walter. These are his cousins down at another estuary called the Breda Estuary. So all the little colorful dots you see are animals that are tagged in that big, the first estuary on the left that you see. That's called the Breda Estuary. And the black dots, if you see below the Breda Estuary, there's a red outline. That is called the De Hoop Marine Protected Area. And what I really want to try and focus on with these individuals that seem to be moving quite slowly is that the colorful ones are moving far further, like far greater distances than the ones that are black. And this is really showing that the animals that are the black ones, they were tagged in the marine protected area, they hardly leaving the marine protected area, where the colorful ones are moving all over the place. So what we think has happened here is that the resident species, the guys that stick around all year, have been fished out of the estuary. And the ones in the MPA, they're safe. You can't fish in there at all. So they're safe, they're protected, they don't need to go anywhere. So they're the ones that are really sticking around. And it really shows how 2 kind of groups of animals living in very close proximity to each other can do such different things. So some blue stingrays, these guys are so cool. So we, back in 2016, I think we started tagging some blue stingrays. These guys are Southern African endemics. You only find them in this part of the world, and they have this brilliant amazing blue mottled color on them. And we tagged 27, and they were monitored for 4.5 years. And they were tagged also very near here. But what was really interesting was that they held very small, they used very small areas. So they hardly moved up and down the coastline. This is just an example of a batch that was tagged down the road, but there was another batch that was tagged somewhere else, and they also didn't move. They seemed to each hold a certain part of the coastline, and they never really interacted at all, bar one individual who, to our knowledge, has been the furthest distance covered by this kind, not the species, but I can never remember if it's undulatory or oscillatory. So I feel like I should know this, but the ones where the wings go like wavy. So this species has recorded the furthest distance moved by this type of ray, which was also really cool. I think it was over 400 kilometers. So it's probably like 250 miles, I think, or so. So it's still an impressive distance for what's probably a smaller stingray species. And what's really nice is that we're actually starting to do more work on an aggregation of these species that is occurring in one of our estuarine lagoons. So every October, the species aggregates in quite large numbers, and we're trying to learn a bit more about why they're there. I mean, we think it's for breeding and for pupping, because we've seen little pups, which are so cute. They're so tiny. But, you know, are they there all year round? Do they leave? Do they come back? And the nice thing is that this estuary is literally in the middle of those two batches that I mentioned now for the previous slide. So we're quite excited to see whether they're coming from both places or what's happening there. Then you get the sharp, bitey, toothy things, which I specifically don't work on, but there's some very brave researchers that do handle things like giant tiger sharks. And we had some colleagues in Reunion Island. They were tagging a whole bunch of tiger sharks and bull sharks because they had that suite of shark-human interactions with quite a lot of fatalities. And what happened was that they tagged a whole bunch of these sharks. They were never detected. There was one tiger shark that was never detected in Reunion, but suddenly it popped up in South Africa, and then again in southern Mozambique, and eventually it got recaptured in Madagascar. So this just shows these huge scale movements of these animals across the open ocean. And the beauty of this compatibility of all of us having the same equipment was that we were able to figure this out, and we were able to pick up these movements, which was really so cool. Okay.
That is really amazing.
Yeah,
I just want to give you a quick little time check. So 5 minutes?
Yes, I was going to say I'm running out of time. So I'm going to skip through this example. It's essentially highlighting how so telemetry can give you such cool information about the movements of animals, but through them being recaptured, we can also figure out some kind of level of fishing mortality. So how many fish are actually coming out the water? And nothing can give it better than this example. So I tagged one little leerie, one of my PhD fish in this Kawi estuary. It was tagged 15th of January 2013. It was 30 centimeters. We know for a fact that it never left the estuary in 300 days because we've got that track. That animal did not leave the estuary. However, we got a phone call to say, hey, you know, we've found this thing inside this fish. So we obviously knew it was dead, because the only way you get the tag out is if the animal has been gutted. But there was a dart tag, so one of those plastic tags with it, and we're like, no, we don't tag our fish with the dart tags as well, like only the tags. So we were like, what is going on here? So we got hold of our friends from that long-term dart tagging program, and we said, listen, we've got this. And they said, oh yeah, that one was tagged you know, a year, so pretty much 15 months after we tagged it with an acoustic tag. But it was recaptured by a boy who was fishing at the river, who released it again. So in this time, this fish was caught 4 times, recaptured 3 times, tagged twice, and killed once. And what's frightening is that all of this happened below, the fish was below the legal size limit to keep it, and it did not spawn once. So in order to, this fish ended up being about 53 millimeters total length, so the whole length of the fish, where it should be almost 1 meter in order to keep it and to spawn. So it didn't stand a chance. So it just shows the vulnerability of these species. So what can you do in terms of telemetry, right? So you've got all this awesome information and we can like give it to managers and do what you want to do with it. But you as general public, what can you do? And it's really managing your catch. So do you need to keep 5 animals if you're allowed to keep 5 animals, or is maybe keeping 1 or 2 okay? Practice good handling, so make sure you handle the animals to the best of your ability, especially if you're going to release it again. You want to make sure that that animal is going to survive when you release it, and educate yourself and others. Like there's so many rules and regulations and whatever, but if you want to know more, like learn about it and tell your friends, oh, by the way, I found out this really cool thing, and this is like the best way to handle an animal or whatever it is. So there's so many other things that people can actually do that will help in the long term. So to end it with, to end everything, I was at a conference a couple of years ago, and a colleague shared this there, and I've always, it stayed with me ever since. So humans are essentially storytellers. And you and me, we're humans, and scientists can be you and me. But by basic maths, we can cancel out some things, and ends up with scientists basically being storytellers. So hopefully you've enjoyed this story this evening. I'm going to do a social media punt. If you would like to follow us on our social media, please give us a little follow. We don't do X anymore. This is an old slide. I apologize. But thank you very much for listening. And sorry to keep you so long, and welcome any questions. Thanks very much, Doc.
This is fantastic. It's such a great example of collaboration and what we can accomplish by working together. It's, that the network you have down there in South Africa is so fascinating. It's great. Such a good research is coming out of it. So kudos to you for putting it in place and managing it and working it to improve it. So
thank you.
Yeah. We have a few questions from our audience tonight, so we can turn to those. And I have some questions too as we go through. You can stop sharing your screen if you want. So first question we had earlier on from Gabrielle. She was wondering, have you ever recorded something like totally unexpected? Maybe not a fish, maybe it is a fish. Or is it usually just limited to certain kinds of marine life? Like, so do your acoustic sensors, do they pick up any other noises besides just the beeps from the acoustic tags, or can you hear like whales coming through every now and again or something?
Yeah, I wish. I mean, if you could put it in all these amazing capabilities in one piece of equipment, that would be amazing. But what we get sometimes is temperature. So we can record temperature, which isn't as exciting as whale noises. But you do get some kind of idea of ambient noise. But what the noise is from, you can't really say. I mean, it might be a boat. It might be a noisy reef. You're not too sure. But yeah, generally you will only pick up the animals that have been tagged. And we generally have a good idea of what is swimming around at any time. Sometimes we get some species that pop up in places that they shouldn't be, generally. And I think that's, with shifting water temperatures and things. So it's getting quite exciting to watch when some animals shift down. Yeah,
that is interesting. I guess when you install a tag or insert a tag, you have some record of its frequency that it emits, and that's how you identify the individual through the frequency emitted to the tag, right? Okay.
Yeah, so each tag comes with like a little sticker on it, and then it says that's its specific tag ID. So you'll know that tag 12345 belongs to a learfish that was tagged on the 31st of December at this place. and then we collate all that information. And then if somebody has receivers and they download their receivers and they go, hey, I've got all these different animals, can you tell me what these animals are, or give me some idea? And oh yeah, okay, you've got 3 hammerhead sharks and you've got 2 of this, or whatever it is. So yeah.
So we had a few more questions just about tagging. But so out of all the ways you showed to different ways you can tag, acoustic tagging and other things, what do you think is the question from Maria Jose was, what do you the most sustainable way to do it? What would your answer be to that?
So I guess depends what kind of angle you're looking at sustainability.
Right.
If it's cost, the little plastic dart tags are probably your most cost-effective way, and they've had real success on establishing citizen science projects, because there are avid anglers throughout the world, and if you are able to train those anglers through little training videos or something how to tag an animal, then they can comfortably go and record all that information. So it comes at very little expense or time or effort on your part. The acoustic stuff, for me, in terms of longevity, so if you're looking at like a sustainable way for like 10 years of data, then acoustics is definitely the way to go, because you can get up to 10 years of data. The satellite tags give you amazing track information, so you can figure out, oh, the animal's literally moving like this, where the acoustic is, well, it's somewhere here and it's somewhere here. But the satellite tags only go for a year, 2 years or so. So it's really short-lived relative to the acoustic stuff. So yeah, I guess depends what your angle of sustainability is. You're
right. Yeah. Ask an acoustic tagging expert, they'll probably tell you acoustic tags are the most
I'm biased.
That's okay. Another question from Aija was asking, do you have manatees in South Africa or anything that's similar to? I don't think you do, right? No. Have you tagged any other marine mammal?
But I guess that's quite far removed. So no, unfortunately not. I think they get higher up, sort of Zanzibar way, so higher up, not Central Africa, but on the west coast, like the higher up you go to the equator, that there are some manatees or dugongs. Again, I know there's a difference between them.
Yeah, I think dugongs are in Australia, maybe. But yeah.
Okay. Yes, it's a spatial thing, but nothing here, unfortunately.
Are there other marine mammals that get tagged as part of the network?
Not of ours, but there are lots of cetacean researchers that are doing things on what our Cape fur seals. So we get a species here called Cape fur seals, lots of dolphin work and quite a lot of whale stuff as well. But we're 2 different schools. But we're hoping to work more together in the future, because I mean, we've got the moorings for our equipment, so we're hoping to allow them to kind of piggyback, so like work together. So do you need to listen out here? Sure, okay, we'll drop it for you and we can maintain it or collect it. So hopefully some good collaboration in the future, but no mammals tagged as such. It starts becoming a ethical thing, because mammals are, if you think of like a dog relative to a fish, the thought is very different, and I think the same applies for whales and dolphins relative to a fish.
Yeah. So I want to ask some kind of like career questions, if that's okay. I know your journey was through Rhodes University and became studied for your graduate studies were there and everything, and now you're a successful researcher. But if someone is interested in getting involved in this field, how might they follow in your footsteps? We had one question from Nicholas kind of related to this, about especially related to IT. It seems like your specialty really relies upon technology, right? So maybe talk a little bit about that and how it might relate to a career in fisheries.
Yeah, definitely. So I think traditionally things were very much hands-on. You had to have you had to be a fisherman and you had to go do this. Very the ichthyology department that I've went through for my undergraduate and my postgraduate degrees is the only one in South Africa. So it's the only place that you can actually specifically study fish research as such. And it was very much always drawing the avid angler, you know, the students, they must fish. I dabbled, but I can hardly call myself a talented angler, but I do try. But I think back in the day, it was more traditional, you know, you did very hands-on sort of stuff, but now, very excitingly, things are progressing and developing where you mentioned IT, you can bring in all these other additional skills if you have, you can combine it with a biological field. So there's so much research at the moment on machine learning, you know, AI stuff, and even maths projects. I've got a couple of maths students. I mean, my maths is awful, but a couple of maths students. And it's data analyses and data management and everything like this real scope to kind of tie the two worlds together. So I guess it depends on, if you're driven, you can find this little niche because they are there. It's just about finding them. And just approach somebody, just say, I've got this cool idea, and maybe somebody will go with it. Somebody will roll with it.
That's a good thing to keep in mind, especially when following your passion, right? It doesn't hurt to ask, right? Just a few more. I know we're getting a little bit long, but I just want to make sure we have some time just for a few more little things on here. So let's see. Oh, quick question. What's your favorite shark? Someone asked.
If you give me fish, I could give you an answer in like 2 seconds.
The Learvis, right? That's your favorite fish. Know that.
We have. I'm gonna have to go with like one of the endemics. I really like the leopard cat shark. So
cool.
It's a smaller species, and if you take it out the water, it curls up into like a little donut, and its colors are really beautiful.
Nice. Someone said that my favorite shark is a frilled shark.
Nice.
Those are really cool.
I like their teeth.
Yeah. Someone asked lightning round now. We're in the lightning round. Okay, ready? Marine organism. What marine organisms show the most surprising or unique behaviors for your acoustic recordings? What did you not expect to see?
Okay, I think the mullet has been really exciting because they're a smaller species, you know, I mean, they're like the size of your hand. And they were moving a lot in the estuaries, which is what we kind of expected. But the number of animals that have been estuary hopping elsewhere, you just go, but you're the size of my hand. How are you moving, you know, tens of kilometers down along the coastline, and then you're turning around and you're coming back? So that's been really cool to see. And then I think excitingly, and I venture hesitantly into this, is the white sharks, because I'm sure that you guys are aware of our white shark situation. You know, we had an incredible white shark population, then 2 killer whales or orca came into the area, and our white sharks have been running scared ever since. So it's kind of been really interesting seeing how the tagged white sharks have sort of somehow changed their behavior too. They haven't, I can't divulge too much because it's not my research, I'm not involved in there, but it's just interesting seeing how things have changed.
Sure. Yeah, that's interesting. All right, so a few more questions. I know that, so the Save Our Seas Foundation funds part of your research. What part do they fund? And you know, you said that, you know, this project has been ongoing for a long time, but how, you know, what are your plans for expanding? How would you expand it to do more?
Well, first of all, shout out to Save Our Seas. They are one of our long-term partners, and it's been an incredible relationship for the past 12 years. So we really luckily have Paul Cowley, who I mentioned. He struck up this relationship, and it's really been a wonderful relationship to be a part of. They essentially, the money we get from Save Our Seas is for running the platform. So without the funding, we actually would cease to be, just because it manages our day-to-day field work, to go and roll over our equipment, to go and get it out, to deploy it, to tag animals, to do all the little basic things that we need to actually run and keep the platform maintaining the platform. So they've been absolutely integral in keeping the ATAP alive. In terms of future plans, hopefully some expansion into the Southwest Indian Ocean. So increase the array in Mozambique. We're going to have some equipment going in the Comores. Madagascar's growing, hopefully linking up with Seychelles. So it could go from a South African array to a real Southern African regional array, which is super exciting. So yeah, we're quite excited about that. And hopefully also venturing into the freshwater side in South Africa. Our rivers are tiny relative to a lot of places in the world, but still lots of cool things to find out.
Yeah, I'm sure. And Save Our Seas does a lot of work in the Seychelles. So it'd be really interesting to use more collaboration, combine all that work together.
Definitely.
So last question for you is just how do we learn more? How do we stay in touch with what you're doing? You shared your social channels, but is there a place we could go and follow your work, or follow what's happening with the array? Are there any like live trackers or anything out there?
Yeah, unfortunately not. That is my dream. My dream is to have like this live portal where you can go, oh, I wonder how many receivers there are, and then you can click on the map and you can see, and like click on cool examples or something. That's like the ultimate dream.
Next year, that's how we can expand.
Maybe the Museum of Science and Discovery can help us.
Yeah.
But that would be the dream. But at the moment, if you do want to keep track, then social media is probably the easiest way. I'll admit that I run the platforms. So I've planned this year. I've planned to be proactive and have created a whole bunch of posts before the time. So hopefully keep some steady content being uploaded. But yeah, that's pretty much the place that you can keep up to date on what we're up to.
Great. Well, thank you so very much for coming in tonight and staying up late just for us, sharing your amazing research and the collaboration, the storytelling. I love the storytelling. That's the best part, I think. And connecting us with South Africa and the acoustic array there. Thank you very much for being here.
Yeah, thank you very much, Lance. And thank you everyone for listening. I really appreciate it. I love the time,
Yeah. Well, with that, I would like to invite everyone to join us next time. You can join us next month. Get rid of my little boxes here. You can join us next month on the 22nd, same time, 6 p.m. Eastern. Dr. Alice Rogers will be here to talk to us all about seven-gill sharks in New Zealand. So we're going from South Africa to New Zealand next time, and how climate change is affecting these species. So you can sign up on the website mods.org/STEM Learning Series, or scan that QR code that's right there, and join us next time. We'd love to have you. Thanks again to the Save Our Seas Foundation for making this whole series possible. We really appreciate their support. Dr. Murray, thank you again. We'll see everyone next time.
Thanks, Lance.
Bye.